Rational engineering of DNA nanoparticle motor with high speed and processivity comparable to motor proteins. Takanori Harashima, Akihiro Otomo, Ryota Iino. DNA gold nanoparticle motor is a burnt bridge Brownian ratchet moving on RNA-modified surface driven by RNA hydrolysis of RNase H, and one of the fastest nanoscale artificial motors. However, its speed is only 3 nanometers per second and still much lower than those of motor proteins. In this study, we apply high-speed and high-precision single-particle tracking and resolve the pauses and steps in the motion of 100 nanometer DNA gold nanoparticle motor. We find that the bottleneck of motion is the long pauses caused by slow RNase H binding at nanomolar RNase H used in the previous study. As RNase H concentration increases, the pause lengths shorten from 70 second to 0.2 second, while 20 nanometers step size are almost constant. At high, micromolar RNase H, the speed reaches 100 nanometer per second. However, we also find that the processivity, run length, and unidirectionality largely decrease. To understand the mechanisms of motion and trade-off between speed and other performances, we also develop a geometry-based kinetic simulation which reproduces experiment quantitatively. The simulation reveals that pauses are caused by multiple DNA-RNA hybridizations between DNA gold nanoparticle and RNA substrate. The steps occur when the number of hybridizations decreases by the progress of RNA hydrolysis. The simulation also reveals that as RNase H concentration increases, the bottleneck switches from RNase H binding to DNA-RNA hybridization. This switch of the bottleneck results in low processivity, short run length, and low unidirectionality at high RNase H concentration. Furthermore, the simulation predicts that large DNA-RNA hybridization rate improves the trade-off. Finally, based on the prediction of simulation, we engineer a DNA gold nanoparticle motor using a DNA-RNA sequence which shows 3.8 times larger hybridization rate than the original one. As a result, the engineered motor simultaneously achieves speed of 30 nanometers per second, processivity of 200, and run length of 3 micrometers comparable to motor proteins. Our study demonstrates a mechanism-based strategy to improve the performance of artificial molecular motors.